So regarding the Human Genome Project, we have to understand what was the main goal of the Human Genome Project, then and what are the major findings or discoveries during the Human Genome Project. So sometimes it is abbreviated as HGP. You may get a question, what do you mean by HGP or what is Human Genome Project? It can be a short answer question or it can be a 5-mark question. It's actually a collaborative international project and that was started in the year 1984, mainly under the leadership of US government. And the project was launched in 1990 and it was completed in after 13 years, that was in 2003. So most of you may not be aware of this Human Genome Project because you might have born during those times. So what are the major implications or what are the major findings of Human Genome Project? Mainly this Human Genome Project was introduced or started to find what are the important genes present in human body. That was the main aim of Human Genome Project. And there was a very famous institute, National Institute of Health. And sometimes when you are studying about NCBI, you know that it is under the control of National Institute of Health. So it's a very prominent health organization in America, and they were funding this Human Genome Project. And along with this government supported project, and this government supported project was mainly a collaborative project. Several other countries were also involved in that, like United Kingdom, Japan, France, Germany, Spain, as well as China. The governments from these countries also offered their support for this Human Genome Project because it was a revolutionary project at that time. But what was interesting that when this project was going on, a private company known by the name Celera Corporation or Celera Genomics has also started their own research, and they were a little ahead of this government supported project. So there was and finally then their data has been uploaded into the public domain. Now it is because of this private participation that all the genome information or gene information that is now available become free for the public. Otherwise that will be a priority of a few countries like USA, Germany. All these participating countries were only enjoying the benefits. Then what was the main aim is that we will be getting an idea regarding the how many genes are present, what are the functions of main genes. So that is the main function, to know about the function of gene as well as the functions of important body proteins. That was the main purpose of Human Genome Project. And because if we know the function of major genes or major proteins, that will be having a lot of benefits or application in the field of medicine, biotechnology, or in other areas of life science. So we will see what are the primary goals of Human Genome Project. And here you can see they are listed into 3-4 categories. The first category is that mapping of human genome. What is a genome actually? Genome means collection of all the genes in an organism. If you talk about genome of an amoeba, how many genes are there in this amoeba? That sum total of all the genes, or collection of all the genes of an organism. So when we say human genome, it simply means all the genes of the human beings. So there is one function of this. One is mapping the human genome. Human genome means all the genes present in human beings. So first phase of this is constructing the genetic map and also to identify and locate the position of various genes. So you know that there are 23 chromosomes are there, and each chromosome will be having some genes. So when we say that gene for a particular cancer, we have to identify in which chromosome, in which arm of the chromosome, or in which area of that particular chromosome this gene is located. You might have studied about Down syndrome, and there is a situation known as trisomy 21. So in the case of Down syndrome, the gene responsible for that syndrome is located in the 21st chromosome. So that kind of information is valuable. So that was what is expected from the Human Genome Project. So and this was done using techniques such as restriction, fragment length, polymorphism. It's a kind of technology used by biotechnologists, and also microsatellite markers. This is the technique they have used for marking the human genome. Now, second aspect of human genome is sequencing. Now you know what is the sequencing. Some of you have sequenced the genes of certain organisms, certain species. So when we say the sequencing, we will be finding out what are the different types of genes present there, what are the different types of codons like thiamine, uracil. That information is what we call by the sequencing. So second phase of Human Genome Project is sequencing the different, if there are 10,000 genes in human being or 20,000 genes in human being, we have to sequence, or we have to find out the FASTA sequence of all these different genes. That is the second purpose, sequencing the human genome. The next phase involved in determining the precise sequence of 3 billion. There are 3 billion. 3 billion means 300 crores of base pairs are there in human DNA. And now earlier it was a big challenge, but now there are automatic machines are there, and these automatic machines will be able to sequence thousands of genes at a time. Then fourth objective is analyzing and interpreting the genome. So you know what is a genome? Genome means entire genes, the sum total of all the genes. So once the sequence was obtained, the data required analysis to identify genes, regulatory sequence, and other functional elements within a genome. If you take the entire genome, you get a sequence of all the genes. All the genes may not be working at the same time, or some genes may not have any work. Some of the sequences were mainly involved in controlling other sequences. That is why regulatory sequence. Some have some other functions also. Some may be inactive. So these are the different types of genes will be there. All the genes may not be working. Only which are the main functional genes, which are the inactive genes. That information we have to find out. Analysis involves collaboration of computational biologists, genetics, and other computational biologists. As I talked to you at the beginning of this topic, that means several classes back. Computational biology is actually an area that involves a lot of computer experts will be there. So then genetic experts will be there, and by other life science experts will be there. All were sitting together, and they will be finding out what will be the possible function of this particular gene, and what will be the function of certain other inactive region, why they are inactive. All this information has to be analyzed and should be interpreted. Then marking the data freely, making the data freely available. Now you may be thinking the human gene. If you want to know about the Human Genome Project, you simply go to the Google and search Human Genome Project. You will be immediately getting the information regarding a particular gene you wanted. That was made possible only because of the involvement of this company known by the name Celera Genomics. Because government of different countries were worried if the information about human genome is becoming the property of few corporations, like a few governments of few countries, and this one private company, they will be making huge profit. So to avoid that, what the government did that they upload the entire information into the public domain. That was why now this information about the Human Genome Project, even though it has been produced using a lot of money, now it is freely available for all the people of the world. So an essential aspect of human genome was ensuring that generated data and tools for analyzing it were made public, fostering, promoting further research and advancement in gene study or genomics. So that was another fourth aim of the human genome project. That is the reason why now we have all this information freely available in the public domain. But there are some ethical, legal, and social implications are there for the human genome project. The privacy and fairness in the use of genetic information, including the potential for genetic discrimination in employment and insurance. For example, now it is not when a baby is born, if the entire genome of the baby is taken and analyzed, sequenced, the scientists will be able to predict that this particular baby has a chance of getting a particular disease or a particular cancer, maybe after 10 years or 20 years or 50 years or 60 years. And if this information is available to insurance company, they will not be ready to give them insurance. So that kind of problem will be there. So that is why every information, even if it is a very good information, can be used by potential people in a very bad, in a crooked way, so that it can harm the public interest. So this is one problem in the use of human genome project. Integration of genetic technology such as genetic testing into the practice of clinical medicine. Because in the genetic, because now it is not very popular, but slowly this genetic testing become more and more popular. Because if you are going for a genetics testing, you will be able to predict what kind of diseases, what kind of cancers, or what kind of other complications I can have after a period of 10 years or 20 years, or after 50 years or 60 years or 70 years of age. And if that information is available, that will be very useful for the doctor to offer what you call prior treatment before even the starting of the disease. Ethical issues surrounding design and conduct of genetic research with the people, including the process of informed consent. Now, informed consent is actually an area coming from bioethics. You are, as for example, when corona vaccine were discovered, few people volunteered for testing, and these people should have. So from these people, the authorities or people who are doing that research should get a type of consent known as informed consent. A consent means an agreement to participate in this research. That is not a consent. Informed consent means the particular person should be thoroughly told about all the negative side effects of this of injecting this vaccine into it. So that is known as informed consent. After knowing the entire negative effects of the vaccine, then he can take a proper whether I should participate or not. So that kind of consent is known as informed consent. We should not hide any information from them. Whatever information is available at that time, it should be shared with that particular volunteer and before starting any kind of treatment or introducing any kind of drug in him. So education, healthcare professionals, policy makers, students, public and the public about genetics. And these are the education, healthcare professional, policy makers, students and the public about the genetics and complex issues that result from genome. Because genes are important thing. Because if you study the gene, for example, nowadays many couples are not having children. If there is a trend in the market, in the hospital industry, that they are selling the genes of certain valuable, highly intelligent people or highly beautiful people, and they are marketing it. Now it is not possible. If such a trend is coming, there will be lot of takers for that. So if I pay this much amount, that I will be having a baby who will be highly beautiful or highly intelligent, who will have a lot of skill, then there will be lot of parents or lot of couples who will be going for that. Such things may not be beneficial for the society. So these are some of the problems of the human genome project. So unless every research has been now moderated in an ethical way, there will be other problems, because it is not the problem of the research. It is a problem of the people who are using that research. The Human Genome Project has approximately 3.1 billion base pairs. That is how much base pairs are available in Human Genome Project. Then it is, as I told you, it is a very important mega project with the participation of several countries and one private company. And it sequences only euchromatic region. In genetics, you have studied what is a euchromatic. Those areas of the chromosome where genes are present are known as a euchromatic region, and which make up of 92% of the human genome. Other regions called heterochromatic region are mainly found in centromere and telomere. Centromere are the center portion, telomere are the end portion, and were not sequenced under the project also. Then it was completed in 2003. Then in 2009, the Genome Reference Consortium released a more accurate version of human genome, but still more than 300 gaps are there, while 160 gaps were renamed in. This is a present status. So in 2009, the information that was produced in 2003 has been refined, but even in 2005, many gaps are still existing. 160 gaps are there. In 2020, the Genomic Reference Consortium has reported only 79 unresolved. That is, still in 2003 there are 2009 300 gaps were there. Gaps were missing information. By 2015, only 160 missing information are there. Now it has been still reduced to 79 unresolved gaps. So that is the present state of Human Genome Project. And what are the key findings? You can see that there are approximately 22,300 protein coding genes. And before the starting of this Human Genome Project, scientists were thinking that more than 1 lakh or 2 lakh genes may be available, may be present in the Human Genome Project. So Human Genome Project was a major work in bringing down that gene number to 22,000. The human genome has significantly more segmental duplication than had been previously suspected. That is, some areas of human gene are duplicates. So that is the meaning of a segment, nearly identical repeated sections of DNA. You might have studied about satellite DNA. So these many areas of human genome are repetition. Why such a repetition are still present in human genome is still not known. At the time when draft sequence was published, fewer than 7% of the protein family appeared to be specific to vertebrates. So there is also some other information about the human genome. Then what are the possible applications of human genome? Human Genome Project through sequencing of DNA can help us understand diseases including genotyping of specific virus to direct. For example, if you know the genotype of any particular, for example, coronavirus, it was because we know we have a lot of information about those type of viruses, we were able to bring a vaccine very fast. Otherwise, normally the discovery of vaccine may take 10 years or 20 years or sometimes 30 years. That was the case with the most of, even if you take the case of AIDS virus, still we don't have a proper vaccine for AIDS virus. So, but in the case of coronavirus, the scientists were able to come forward with a vaccine within 1 year or 2 years. So that was a major achievement. All this because of the genotyping of virus genome. Identification mutation linked to different forms of cancer. Why certain cancers are developing in particular individual. That is also an important advanced achievement. Design of medication or means medicines and more accurate prediction of their effects. So when we are taking one particular medicine, what are the effects, good effects as well as bad effects of that medicine in our body? Advancement in forensic applied science, biofuel and other energy applications like agriculture, animal husbandry, bioprocessing, risk assessment, bioarchaeology, and because basically this human genome project contains information about gene. That information, for example, if there is an unknown baby and police discovered that one unknown baby, very small baby has been found from somewhere, when if that news is available, there will be a lot of people who will be coming to police. That is my baby, give it to me. But police will not give it. What police people will do? They will take a DNA fingerprint. And such information are possible because we have this kind of technology now. And the proposed benefit is that commercial development of genomics research related DNA, because this is also a because drug industry is one of the biggest industry in the world. Even if people are dying, this drug industry because of corona vaccine. We know how effective that corona vaccine is now because we are all experiencing it. But still, at that time, there was one company, I think Madonna Genomics or some, that is the name of that company, that was started in USA. Now, if you go to the value of that company, they are a multi-billion company. So, by selling this vaccine, they made huge profit. So that is how this drug industry is working, especially in European and American or in most of the Western countries. Even if you go to Canada, USA, any European country, the most important problem you will be facing is getting a proper treatment. Because without enough money, you will not be able to go to the hospital, and even consulting a doctor may be very challenging there. So the sequence of DNA is stored in database available to anyone in the internet. So it is now publicly available. US National Center for Biotechnology Information has gene sequence in the database known as GeneBank, along with the sequence of known and hypothetical. Already we have been working with the GenBank. So we know how GenBank is working. And several other organizations like UCSC, Genome Browser, the University of California, Santa Cruz, Ensemble, they will be also giving additional data about that. And computer programs have been developed to analyze the data because data itself is difficult to interpret. Because if you simply go see a faster sequence, human being may not be able to get any information. Only with the help of certain computer tools, software programs, we will be able to properly analyze and get the required data. For example, just now, if you take a spider species, that FASTA sequence is not giving you any information. Only if you put that FASTA sequence in the GenBank and do a blasting of that, you will be able to get the valuable information from that. So that is why without computer technology or software technology, it is impossible to properly analyze the human genome information. So this is an idea regarding what are the major achievements in Human Genome Project, why it has become a public property,